Hello and welcome, everyone. We're giving everyone a few seconds to join from the waiting room. And I think we can start the webinar. This is Sabrina Paganoni. I'm joining from the HEALEY Center, HEALEY and MG Center for ALS and Mass General. And today we have a fantastic group of speakers and panelists. So really, really excited to be here. If we can go to the next slide. So today we do have our weekly update, but really the reason we are so excited about today's webinar in particular is because we're going to be talking to different people who will bring a diversity of perspectives about participation in the trial with respect to lumbar punctures. As you know, for those of you who have been following us on this webinar series, we really are emphasizing the importance of biomarkers as an integral component to this trial, and some of those biomarkers can only be measured in the spinal fluid, which is why lumbar punctures are so important. So I just wanted to introduce our guest panelists and speakers for tonight. And first of all, I'm really excited we have Mr. Steve Witt, a person living with ALS who will share his own experience with lumbar punctures. And so thank you so much for accepting our invitation. We really will be hearing from you first. And just, before we go to you, I just wanted the audience to know that today I'm here with a few colleagues as well, Dr. Cudkowicz, whom all of you know well from this series, as well as our colleagues, Dr. Lada and Dr. Driss, who have also already been guest speakers on this series, and they are lumbar puncture experts. So that's why they're here tonight to talk with all of you and Mr. Witt as well in terms of, kind of different perspectives in terms of lumbar punctures. And then, as always, we have Kathleen Small and Alison Bullard from our patient navigation team. So next slide. It is truly a pleasure for me to introduce Mr. Witt. I had the opportunity to meet him in the context of the ALS diagnosis. And I had to say the favorite part of my working with people living with ALS is that I get to meet special people and special families like the Witt family. And so thank you so much, Mr. Witt, for being with us today. I'll let you introduce yourself. I just want to say that I've learned so much from you personally, also as a physician. You teach me a lot about ALS, and I know you'll talk about how and through your writings, which are fantastic. And so I just want to give the floor to you, and thank you for being here tonight to talk about your experience as a trial participant.
Sure. Thank you. Thank you, Dr. Paganoni. I'm happy to share my experience with lumbar puncture, but also more importantly, talk about why I enrolled, despite feeling like some of you may, a certain hesitancy about that procedure. First, you should know that I have a fairly low tolerance for pain and a healthy suspicion of anybody approaching my skin with a sharp object. During blood draws, for example, I not only avert my eyes, but also close them to ensure I remain absolutely oblivious to what's going on around me. So right there, that's two advantages that lumbar puncture has for me. There's no blood, and it's all done behind my back. For those unfamiliar with the procedure, I'll describe briefly what I experienced that day. And if I had to put into one word to describe it, that word would be uneventful, which is something one dearly hopes for any medical procedure. So in mine, first the administering nurse gave me the choice of her describing each step as we went along, or I could listen to music of my choice, or just sit in silence. I sat on the examination table, rounding my back and resting my elbows on a couple of pillows. The nurse sat behind me, and I felt a mild pinch as a lidocaine injection numbed the area that she had marked carefully for insertion. Then the hollow needle was inserted, and only once did I feel slight pressure during this process. The spinal fluid was extracted, and that was really it. I'm told that the new needle they use with a beveled edge has significantly reduced the number of side effects. I mean, I can't say you won't experience any, but I certainly did not. I stayed hydrated and I did not experience any. But listen, if you already have the courage and determination to take that awful Relivial every day, twice a day, believe me, you can handle lumbar puncture. I was diagnosed a little over a year ago when only one oral therapy was approved. Now there are 3. People with ALS, people like us, agreed to be in those clinical trials. And because of their commitment, we're here potentially receiving the benefits of more months of life and improved function. Now, you can call it playing it forward, but I call it a simple debt I owe to them and to future ALS patients to do my part to advance the science. And when I see the passion at my ALS clinic to finding a cure, I think I owe them too. That's why I also participate in 4 observational studies, 3 at MGH and 1 at Emory. That's really all I have for you on LP, but Dr. Paganoni said I could have a moment of shameless self-promotion and mention my journal, No Leg to Stand On. Surprisingly, there was much about this disease that I found unnervingly funny. Dark humor, of course, but when I started writing, it was for a small audience of friends and family, but it soon became something more with hundreds of readers in the ALS community. It's No Leg to Stand On. Please take a look. I'd love to know what you think. So send me a comment. Thanks for your time, and I hope this has been useful for you to make a decision.
Thank you. Thank you so much. And I also see here that in the Q&A box, another participant in the trial also shared their comments about their own lumbar puncture being, uneventful, as you described it, no pain, no pressure, with the nurse and doctor making it very comfortable. So again, it's great that as a community, people living with ALS are also sharing their experience, as you are doing, as the person who wrote in the chat is doing, and as you're doing with your journal. I think we all learn from each other. So thank you so much for that. And if anyone has questions for Mr. Witt, please put them in the Q&A box and we'll definitely ask them during the webinar. While you are typing, I do want to move to the next slide and also introduce Dr. Driss and Dr. Lada. I'll just turn it over to you. I know you you see kind of, you do lumbar punctures all the time. You're the people performing them and working with people with Mr. Witt who are, generous participants all in the trial. So do you want to share your thoughts? And I think Mr. Witt described the process really, really well. And so I want to see from your perspective of doing it, what are your thoughts? Who wants to go first? Oh,
Senda, there you go.
I started talking and I realized I'm muted. No, I really echo what you said. Every time when I describe, when I try to consent a patient for a study and we get to the part of the lumbar puncture and I describe what it is and what we need and why, I never have a patient tell me no. They say yes. I mean, they're not hesitant. I think a lot of, as Shafeeq said earlier, maybe some of the hesitation is the way we present things to patients. So far, I have enrolled a few patients. They all had lumbar puncture and eventful, thank God, no problem. And nobody had any problem after with post-LP headache or anything. So things have been working really well so far.
Yeah, I would echo that. I mean, I think having a conversation with your physician about the real risks and benefits is really important. I would point out that if we look now at some of the drug development we've had, some of it we greatly owe to the lumbar puncture. So, I mean, I think tofersen is a great example of a drug that really was approved because of its phenomenal effect on neurofilament levels. And really what we know about neurofilament initially came from our understanding through spinal fluid. We then refined the technique so that we could measure it in blood more easily, but we may have never really known about the utility of neurofilament if we had not discovered this through spinal fluid in ALS patients. So this is not just theoretical. This really, I think, means something and really helps us in the drug discovery process. At our institution, we've been doing lumbar punctures on ALS patients for now, probably close to 15 years. And we've done literally hundreds on patients, some of whom have had 10 or 20 lumbar punctures serially over time. And I think that we've had maybe one person who's stopped getting lumbar punctures because of the procedure itself. So it's really an extremely well-tolerated procedure, despite what I think the public image of it is. I often, as I talk to my patients about it, I guess I'm in the fortunate or unfortunate position of having had 2 lumbar punctures myself. And so I often discuss that with patients and tell them about my experience. And I think to some degree, that gives them some comfort that, number one, I'm not doing anything to them that I haven't gone through myself. And number two, if wimpy old me can do it, they can do it too. So, I really encourage people to really talk to their trialist about this. It's really important data that we get, and really in the end, it's an extremely well-tolerated procedure. And the more people that have it, the more they can talk to other people and tell them that they didn't have such a bad time with it. And really, this is something that could exponentially benefit the ALS community.
Yeah, I think it's very important what you said about how much these procedures have really helped the field. As you mentioned, the case of neurofilaments, which initially were only measured in the spinal fluid, and now we can measure also in blood. And so that's actually what we want to do, especially with the current regimens. There's a lot of very new biomarkers that can only be measured in spinal fluid for now. And by collecting the fluid and analyzing it in the platform trial, we can help test these drugs, but also really develop something that could help everyone. And in fact, as we were talking, somebody else commented in the chat in terms of having 2 LPs performed with no issues. So thank you to all of you again. Thanks again, Mr. Witt, for sharing your own experience, and thank you to the people that are writing in the chat, so that the more you can talk to others, I think the more we can make progress. And we do have some materials on our website. So if you can go to the next slide, I believe that we can see here actually Dr. Shafeeq Ladha in the picture there with Alison. They were on a webinar here, and when they presented the video that you can find online, please check the QR code. Dr. Lada made this video, and so thank you so much, and came on our webinar to answer more detailed and technical questions about the procedure. So don't miss those resources. And also we have additional brochures and other resources on our website. So if you want to again learn more about the technical aspects of the procedures, we have those materials available, and we'll continue this conversation, or you can ask questions now as well. Next slide. And I also wanted to mention that, again, we're very excited about the regimens that we are enrolling for right now, regimens F and G. Those are these new, brand new drugs, and we really important biomarker information that we're trying to really gather to support their development. And really thank you to all of you who are participating. We're enrolling actively for these regimens. And if you're interested actually in participating in these regimens, if you can go to the next slide, I want to share some more information. Again, we're enrolling for 2 regimens, the F and G testing drugs made by Cudkowicz and Denali respectively. Next slide. And you can see here that now we have many sites active. They're all listed on our website. For example, the Barrow Neurological Institute, where Dr. Lada is the PI, is recruiting. And so if you go to our website, for example, you can see the contact information for their site. There's many other sites recruiting, as you can see here. And the QR code will bring you to their link and contact information. Next slide. Last but not least, thank you again, Catherine and Alison, for being our patient navigation team and always being available to talk to our patients. We're going to have another guest speaker next week, Gwen Peterson, who many of you know, a fantastic advocate for ALS research. She'll come and share her experience with ALS research as well. Then we're going to have another webinar with no guest speaker in August, and then one will be canceled due to travel. So these were the slides. I think we can pick off the slides and have a little bit of a discussion. And feel free to type any questions that you may have. Any questions from the panelists for Mr. Witt, or do you have any questions in terms of, the lumbar punctures?
I don't know if this is really not a question, but I just wanted to thank Mr. Witt for coming and sharing his experience, and in particular, I mean, I think I speak for all of the ALS clinicians here that when we see someone who, despite having this terrible disease, can continue to have such a zest for life and continue to try to find a humor and joy in things, that I think that's really warming to us too, and that's really our goal in what we do. Obviously, we want these medicines that we're developing to help cure or prolong people's lives, but ultimately right now we're really focused on quality of life. And so it's really nice to see that you're trying hard to continue having quality of life. I applaud you.
And that's why I love reading the blog and your journal. I had to say I don't miss any issue, and thank you for sending it to me. I wish I could write like you do. You really can connect to the audience. So.
I have one question for Mr. Witt, if that's okay, Dr.
Sure.
Paganoni?
Yes.
Okay. So we often all day talk to different people and potential research participants, and we do hear a little bit of anxiety over this procedure sometimes. So I'm wondering, did you feel that at first? And if so, what made you kind of change your mind and understand a little more, or did you just have that zest for I'm going in to start with?
Yeah, I mean, like I had said, I thought I owed the ALS community and those who had studied, were in studies before, to participate. All I knew about I knew Spinal Tap, and I knew bad hair and bad music. So of course I associated some of that with that, and really the people who I knew who had one had them as a child, and that was frightening experience for them, and that's what they relayed to me. So I just, whatever trepidations I had, I overcame with reading about it, and then just, diving in, and, it all worked out, so.
And I do want to emphasize one important point, like, the techniques have evolved over time. And in fact, we use special needles as part of the trial. And so really what people can read, if people read or learn about experiences from the past, I mean, that was probably with different needles and different techniques. So I also want to emphasize that point, that we use needles that have been specially developed to be essentially more user-friendly. And Dr. Cudkowicz, do you want to tell us more about biomarkers and the exciting science that we're trying to really contribute to by analyzing spinal fluid here?
Yeah, thank you. Yeah, I wanted to I also thank Steve for coming. I'm going to be reading your blog this weekend, so thanks for the link. It's really what Shafeeq said. It's because of people who have been part of these biomarker studies and donated their blood and spinal fluid that we have so many new discoveries. You know, the neurofilament is one of them, but now there's research finding things called cryptic exons in the spinal fluid and markers of inflammation that are really telling us more about the biology and helping us with different drugs and see if we can tell if they're working faster. So it's almost like now the technology and science is caught up with what we can do with these samples and having those samples in these repositories that Niels has and continue to replace those with new ones is just moving the field faster. So I'm very grateful to people who are doing this. And there's so many other ways. If somebody doesn't want to do a spinal tap for a research study, there's other ways that people can help, whether that's blood for DNA or digital markers. There's many ways that we can all work together to figure this illness out.
Yeah, well, one question I have mostly for Dr. Driss, Dr. Lada, you do spinal taps all the time. So the potential discomfort or the anxiety, could that be dependent on the practitioner's experience? And can you tell us more about really the level of rigor and training that everyone who's part of this trial goes through in order to be able to perform the lumbar puncture?
Well, if one of the things that you really want is clear spinal fluid that is not contaminated by blood, because then you're doing a blood sample basically, and especially for biomarker. So people who do spinal tap for research are usually people who are trained and have done many spinal tap before, because we want to make sure that it's one tap and they get the fluid and done. They're not going to be trying to learn or any of that. Of course, I mean, if a resident is just their first spinal tap, it's going to be different, but these people will not be there doing spinal tap for research because the quality of the fluid is important. And as Sabrina and Shafeeq just said, the needles are thinner, they're less traumatic. So technique had evolved over the years, and the more experienced person doing them, the more kind of luck they have, and the less side effect the patient may experience. You have to understand, if a young patient may tend to have a little bit more headache than an older patient, just because of the physiology. When we are young, our brain are a little bit bigger than when we get older. So sometimes that may happen. But again, the very thin and new needles hopefully avoid that.
I would add, I think that, needles create some sort of an almost automatic psychological reaction. And we see this in the EMG lab as well. Yeah. And I think experienced lumbar puncture physicians are also good at alleviating that psychological effect. And so, we try to converse with someone while we're doing the procedure, ask about their life, what are their plans this weekend. I think all of that really has an effect on making the person undergoing the LP more relaxed and maybe a bit distracted even, knowing that they're realizing that you're not anxious as well as you're doing the lumbar puncture, because I think feeling like the person who's doing it doesn't know what they're doing is also psychologically bothersome. So I mean, I think there's a lot of techniques during the lumbar puncture, in addition to just performing a good technical lumbar puncture, that you can use to keep people sort of calm and relaxed.
Yeah.
I don't use, sorry, I was just going to say, many people ask about, do you use sedation or do you knock people out? And I think most of us do not. I mean, obviously for the little babies, when they have to get lumbar punctures, they sedate them. But for adults, I think just sort of relaxing as much as possible is all that's necessary.
Yeah. And as we're talking, we got a few messages in the Q&A box saying that, again, many people share that it was important for them to contribute to ALS research and find new treatments, and that was why they were happy to do the LP. And also other people have commented that both as part of this trial and also part of the tofersen studies, they've been getting a lot of lumbar punctures, because also, you know, for tofersen that's needed, and so that they've had a good experience. So thank you to all of you for sharing all of this. So a few more questions. Dr. Cudkowicz, can you tell us more about progress in terms of the current regimens? I know we're still screening. How many spots do we have left? And then any updates on regimen E as well?
Yeah, thank you. First of all, there's lots of spots left. So if people are interested in being part of the study, please reach out to Catherine on this call if you want help finding a site, or obviously your neurologist, or look at our website. I believe right now there's probably between F and G about 140 people in the study, and we're looking for I'm trying to do the math, but over 300.
Yeah.
So exactly. There's many spots. So thank you. It is enrolling well, which is really good, and we have more and more sites activated for both. So I will say the faster we can finish enrollment, the faster we get the results. So in the fall we'll be talking a lot more and to try to get enrollment to finish soon.
That's a good question about the importance of biomarkers and drug approval. So biomarkers can be used for different reasons. So Dr. Cudkowicz, can you tell us more about sort of, how do biomarkers help with drug approval, and do they correlate with functional measures or disease progression?
Yeah, there's many ways we can use biomarkers. So the one most tied to drug approval is what happened with the gene therapy trial, tofersen with the SOD1 for people with SOD1 mutations, where neurofilament was used as a biomarker for accelerated approval. So it was used as a biomarker of treatment effect. So in that study, the neurofilament level went down by 50% at 12 weeks, and then there was a clinical benefit that was seen a little later. So that's called a surrogate biomarker, so something that predicts clinical response. And that is very exciting. That's probably one of the most exciting things that have happened this year, besides having obviously 3 new drugs that we can be offering our patients. But that is a game changer for the field. Now, biomarkers can also be used to help with diagnosis, maybe help with prognosis. So there's many different tools, the ways that you can use biomarkers.
And one question, when we talk about inflammation, is that similar to the inflammation in multiple sclerosis, or are we talking about something else?
I'm happy to try to answer that. So I say inflammation is a very, I guess, generic term. It means that there's some part of the immune system that might be activated, and that's a very complex system. So I think there's some overlaps with the inflammation that we see in ALS with MS, but not 100%. There's different parts of the immune system involved in these 2 illnesses.
Yeah. So there's one technical question that I'm happy to take. There was a question about, on occasion, and that's rare, but it can happen. Sometimes it's difficult if the lumbar puncture is attempted, sometimes due to different reasons, perhaps anatomy or other technical reasons, the collection may not happen. In other words, very rarely, but it can happen that, the procedure is attempted, but it's not successful. So depending on the regimen, and that's the question in the chat, actually, sometimes it makes sense to do, we generally do the lumbar puncture at the beginning of the trial and at the end of the trial, and to see kind of a change. So depending on the specific regimen and the specific biomarkers, it may make sense if it's missed at the baseline visit to still do it at the end of the trial, but for other regimens, just scientifically based on the specific biomarkers, it may not be needed. So I just wanted to thank the person who asked the question and for volunteering, even if I understand it was not successful in that particular case. Again, it depends a little bit on the specifics of the regimen. Each regimen can be a little bit different with it, but thank you for trying and for volunteering. And I also want to thank everyone who put a lot of great comments again about their own experience with lumbar punctures. Again, all positive notes. So thank you to all of you for sharing that. And before we end, any from my colleagues, I wanted to ask you any additional comments about lumbar puncture and biomarkers?
Healing takes time to occur, so maybe running short trial is important because you want to put patient on actual drug very quickly. LPs and biomarker study are even more important nowadays than they used to be before, when we could like run a whole year or a 18-month trial. Just keep that in the back of your mind.
Good point. As we develop new biomarkers, we could actually make trials even better. Yeah. And questions about HE, Dr. Cudkowicz, I wanted to ask you something in terms of, can you tell us more by regimen, sort of, whether it is required to have lumbar punctures to participate in the trial or not, and a little bit of the differences there?
Yes, so it does depend by regimen. So for regimen F, it is a requirement, and that is because it's so important as a way to tell if people respond to the drug, as well as if there, there's variability in that response, and that predicts a clinical response. So for that one, it is. For the other ones right now, it's optional, but we really encourage people because it's so enormously helpful. So even for G, while it's not required, we are asking participants and asking our sites to really try to do this as much as possible.
Thank you so much. And before we end, I do want to give the last word to Mr. Witt. If you have anything else that you want to share with us, we are here, and thank you again for participating.
Sure. I think I was one of the first people to enroll in my regimen, and I'm approaching 6 months, so I'll be getting another lumbar puncture in September, and not worrying about it at all.
Thank you.
Sure.
Thank you so much. And thanks everyone. And please check out the resources we have on the website, including the video that Dr. Lada made and his webinar with more technical notes about lumbar puncture. So we have lots of resources, and we can bring this topic back to this webinar, over the next few weeks in case you have additional questions. So thank you so much. And next week, we'll give another update on all the regimens with more information about timelines and enrollment numbers. Thank you so much.
Thanks, everyone.
Thank you. Thank you.